So what kind of biological information is available for us to connect? And there is a ton of it. I know you know a bunch of this, but I always think it's good to review. I'm sure you've all heard of the central dogma of biology. This is the holy grail. We used to bow down to this, 20 years ago. It's not necessarily as accurate as it was, but I mean, the basically kind of holds up. You have DNA. So in a human, we have 6.4 billion nucleotides. They have genes within those that can produce messenger RNA or copies of itself. There's about 25,000 genes in the human genome. From here, this messenger RNA is translated into proteins. And these proteins, as far as humans go, are just in its purest native state are about 500,000. We can measure all this. And just this information alone is a ton. But it doesn't stop there. Right? Anybody here working on microRNAs? No? You will. They're little pieces of mRNA, and they basically, again, it's another layer of, they will bind the messenger RNA and cause its destruction. It's another layer of control over the biological processes. And again, we can measure this, and we like microRNAs because they're small and they can be found in the bloodstream. So that's, again, another thing we can measure. Even the messenger RNA, there's not just 25,000 messenger RNAs, that this messenger RNA has introns and exons, and it can be shuffled and manipulated. I used to work on a gene that had 11 different isoforms of itself, and they all did something different. It would drive me crazy. But again, here's another layer of complexity that we can measure. Okay, proteins. I used to do proteomics, and I got the heck out. It's tough. Proteins are a mess. You can do anything to your protein. You could ubiquitate it, put phosphorylation, methylation, anything you want. Again, we can measure all these. It'll make the protein do something different. Let's go back to the DNA. The DNA itself, it's easy to think of it as just straight up sequence, but it isn't. It's constrained by structure. And that if I took the DNA out of one of my cells and stretched it lengthwise, it would be as tall as me. If I took all the DNA out of my body, it would go to Pluto and back, right? For DNA to fit in your cell, it has to be compacted, and it has to be compacted very tightly. And again, histones are very important to that. We can measure that, that condensation of the chromosome. Again, you can methylate histones. You can cause areas to be tighter. You can acetylate them and cause them to be basically looser. Again, we can measure all this. Now, why is this important? How's that? Yeah, you can kind of see that. So these are two insects here that I show. One is a locust, and the other is just your regular grasshopper. This locust is shorter than a regular grasshopper. It's a different color. It behaves differently than a grasshopper. It's more aggressive. It'll get with its buddies and fly thousands of miles and go to communities and eat all their crops, right? Grasshopper, green, a little longer, pretty mellow, kind of stays local. The only difference between these two insects is the way their DNA is packed. Their DNA sequence is exactly the same, right? I would say condensation of the chromosome is actually the most important structural element or control that we have as far as gene expression. But again, all of this stuff we can measure. We can use all of this stuff to figure out what's going on in, say, a disease, an organism, you name it. But what we mostly do is look at my bread and butter in bioinformatics is looking at mRNA. Again, DNA has genes in it. The genes want to do anything in the cell. It makes a copy of itself. This copy goes to the ribosomes for instructions to create a protein. It's almost like headquarters sending emails to the factories to make product, right? Most of the time, do the workers always do what the boss says? You guys have talked to the wrong group. Everybody does what they... No, they don't, right? But most of the time they do. And so that's why we think of this as what we do is we hack the cell. We basically hack the email and we steal all the emails of what's going on between the headquarters and the factory. And based on those emails, we can kind of figure out what's going on in the cell, okay? And what's changed the entire landscape of all this is gene chips. And I brought some, I think. Yep. I'll pass these around. This chip right here has changed biology completely. That I can measure the gene expression of every gene in a human on this chip, in you. And we use this a lot. This has changed everything. Before this happened, we had to measure genes separately. Like, I wanted to investigate this gene, so I'd do a RT-PCR, and then, oh, well, maybe I'll see what it does with this gene, and I'd have to do another RT-PCR. We can do all of that on one chip at one time. So on those chips on the back, what we do is we isolate total RNA. What we can do too is, what's nice about eukaryotic system is they have a poly A tail, so we can actually only isolate the messenger RNA, which helps us kind of reduce, what we have to test. We basically make lots of copies of it, but when we make the copies, we add some biotin. We basically add a fluorescent element, chop them up, inject them into these gene chips, and through about 16 hours of hybridization, those sequences on our mRNA that are labeled will bind to its exact coordinates on that chip. Then we wash off whatever doesn't bind. We take this very expensive scanner, and we basically can see a gene that is higher will have a higher fluorescence. And then we assign numbers to those, right? If this color is white, this will be a higher number, this will be 10, and this lower number or this lower fluorescence will be 2.